Oh, Mark, you have a minute? I got a question for you.
Yeah, you know, I saw you in class with that nature, and I'm not sure, but I think during part of the lecture you were actually reading that.
Well, I was reading your article in here, Mark.
Oh,
that's why.
well,
I
as
mean,
long as it's that, okay.
I think it's
well,
fair.
that's worth it.
But I did have a question about class. You gave the class a problem on phylogenies. It had to do when we were dealing with horizontal gene transfer.
All right.
You had this phylogeny up here, hypothetical species A through I, and then you were talking about horizontal gene transfer and how it might influence a phylogeny.
Correct.
So you had the origin of this unique gene being on this branch going up to A and B. Okay?
Right.
It was then, a copy of it was transferred over here to this site on the branch. And then, of course, that gene would be propagated, you know, up through H and I and up here through G. Then you had a second horizontal gene transfer in which the gene was copied and transferred from branch G over to branch D.
That's
correct?
Exactly right. Exactly right.
And then you ask the class, if you're looking at this unique gene, what sort of phylogeny would be produced?
Right.
Now let me show you what I produced. I mean, I thought to myself, well, what species will have this unique gene? Well, it'll be A and B, it'll be D, it'll be G, it'll be H and I, because they got it in this direction by vertical gene transfer.
Right, yeah.
And the ones that won't have this unique gene are C, E, and F.
That's right, too.
So let me show you the phylogeny that I drew up.
Good start.
I thought the phylogeny should have been branched this way, and then drew C, E, and F. And then I drew it this way, and then had 1, 2, 3, 4, 5, 6 of these, A, B, D, G, H, I. And my criteria were pretty simple, simple to what we did in the primate lab this week, like having a tail or not having a tail, having a gene or not having this unique gene. And so here was gene absent, and over here all of these was gene present. This unique gene, this unique brown gene right here. So I'm just wondering, what's wrong? I mean, just like we could use lack of tails to be the great apes and having a tail to be the monkeys, this is what you ask in class. Yeah, you got something completely different. Could you help? I will sleep much better this weekend if I know the answer to what's going on here.
Wow, that's a very thoughtful analysis, and there's nothing incorrect with what you did here. I think the part that perhaps got lost is how you treat a trait, especially a genetic trait. So indeed, if you're making a phylogeny, you can use the presence or absence of a gene, a total gene, as a character, just like tail present, tail absent. But the thing about genetic information is it includes so much more. It's not just is the gene there or not, but a gene consists of thousands, potentially tens of thousands, of individual base pairs, those steps in the ladder of the DNA. And you can use the very specific information. So if you say gene present as a character, yep, they just all got it. And there's not much more you can do. But if you look at the actual gene, the message, it's a message. It's a set of instructions. And those instructions change through time, and that's what allows you to tease these apart. So use the example of tail, and tail present versus absent. Sure enough, you can use that. But there's tails and there's tails. It turns out that the New World monkeys, Lagathrix and Alawata, have what are called prehensile tails, so they can grab onto something and hold. And monkeys in the Old World, like the macaque, they can't do that. So you could take tail and divide it into a finer category. Can I grab or not? And that would actually be a phylogenetically useful trait. The same with the DNA sequence. The thing to keep in mind, say going back to here, so the gene plunks into this common ancestor. All three of these have it by vertical descent. But you need to remember that as this gene is being passed, this is potentially thousands, millions of years, millions of generations, this gene is changing generation by generation. That's evolution. So it's not the same gene here as it was here. And since this common ancestor, the gene changes here versus here. So what biologists can do with DNA sequences is line them up and count the many, many changes that would accumulate over long periods of time. So I can tell. I can tell that A and B, their version of the gene is pretty similar to each other because they've only been apart this long. And I can tell that the whole set that came from this transfer is distant, more distinct from AB than all of these put together, etc. If I look at D, it'll look quite close to G because it transferred after this split. Therefore, you get the alternative phylogeny that we had eventually in class.
So I think I'm getting it now, Mark. So I guess what I wasn't taking into account that we're dealing with time. Right. The top is time. And as time goes on, the sequence of this gene is changing.
All the time.
All the time. So gene absent and gene present, that's just one data point, a yes-no answer. A gene is not a yes or no answer. A gene is 3,000 bases. Each of those 3,000 places could change as we go through time. And the more we go through time, the more of those 3,000 places will change.
Perfect.
Okay, I wasn't making that connection.
So the thing to keep in mind to construct the correct phylogeny is that the greater the time since divergence, the more different the DNA sequence is. So if we go back to this overall phylogeny here, remember there were two transfers. We'll have to keep track of them separately. But this gene right here represents the last common ancestor of all of this gene in any organism that has it. So if you think about it in this way, what copies have been apart the longest? It's right here. These and all of those that transferred over here have been apart since that first transfer. So if we were to start at the bottom and say, what's the biggest branch? There would be a branch here that goes to A and B, and another branch here that represents transfer number 1. So this is horizontal transfer 1. And interestingly, it doesn't matter what organism this gene sits in. As time passes, whether it's on that branch or that branch, changes occur. Here during vertical transmission, here after a horizontal transfer. So now we've got this lineage of that gene going to GHI. So GHI also appear here in the same order because that gene is being passed vertically, splits, changes on that line, changes on that line, G, H, I. But one more transfer happens. This same G line version moves over to D. But look how recent that is. This is a very small segment of time for them to diverge. So that second transfer is shown here. Horizontal transfer 2. But that's so recent that the D version and the G version are very similar to each other. So they share a common ancestor. The D is on the G line. The G is on the GHI line, which is the most distant from the original that went up to the A and B by vertical transmission.